Hello everyone, this is Dr. P. Mary Anupama, Head of the Department of Biochemistry, St. Joseph's College for Women, here with the topic Coordination between Nervous and Muscular Systems. The contents of the lecture will include Neuromuscular Junction, followed by Excitation, Contraction Coupling, Mechanism of Muscle Contraction, Muscle Relaxation, and Integration of Neuromuscular Systems. We all know that nervous system, it detects the stimuli and it generates impulses in the body very quickly. These signals, they travel through neurons, they reach the muscles for coordinating various responses throughout the body. This neuromuscular junction, it transmits impulses from the neuron to the muscle fibers at a synapse region, which we call it as the motor end plate. The neurotransmitters like acetylcholine, they trigger the muscle contraction. So muscle contraction or even muscle relaxation, that produces the muscular movement. And here there's a coordination which enables the reflex or the voluntary actions efficiently taking place with precision with the trigger of the neuron. What is neuromuscular junction? This is also called as the motor end plate. And this motor end plate is the neuromuscular junction which is specialized in the synapse formation between a neuron and a muscle fiber. So this connects the axon terminal or the axon knob with the membrane of the muscle called as the sarcolemma. This muscle region which is involved in formation of this junction, we call it as the motor end plate. This we call it as the motor end plate. And here we have the neurotransmitters as acetylcholine. So obviously the motor end plate, which is the postsynaptic membrane of the muscle fiber, it has receptors for acetylcholine. This structure, it ensures that the signals, the nerve signals, they travel efficiently to the muscle. Now let us see the events at the neuromuscular junction, that is transmission of nerve impulse across the motor end plate. The action potential as it reaches the axon terminal to this knob, axon knob, which is the presynaptic membrane. So as it reaches down, here you see that the voltage gated calcium channels, they open up. As they open up, there is rapid influx of calcium ions taking place at this axon terminal. So many calcium ions move into this axon terminal through the voltage gated calcium channels. Now, because of this, the synaptic vesicles, which consists of acetylcholine, they are dragged to the presynaptic membrane. They move to the presynaptic membrane. They fuse with the presynaptic membrane, releasing acetylcholine into the synaptic cleft. So here acetylcholine is released. Next, what happens is this acetylcholine, it will travel through the synaptic cleft and reaches the postsynaptic membrane. So here, this is the postsynaptic membrane of the muscle, which consists of nicotinic acetylcholine receptors. These are ligand-gated sodium channels. So once the acetylcholine binds to this membrane, it becomes permeable to sodium ions. So as the channels open, there is rapid influx of sodium ions taking place, initiating depolarization. So here you see that end plate potential is generated. Once the action potential is generated, now it is time to clear the acetylcholine which is present in the synaptic cleft. This is because of the enzyme acetylcholine esterase. It hydrolyzes acetylcholine, restoring the membrane potential here. And now later what happens? Here the action potential which is generated, it has to be transferred down. So what you see is that here as the sodium ions influx is taking place, action potential is generated and it is transferred through the sarcolemma because of the T tubules into the sarcoplasmic reticulum. So the propagation of the action potential, it goes deep down into the sarcoplasmic reticulum, which stores calcium ions through the T tubules. So once this action potential is transferred here, it triggers the T tubules to release calcium ions from the sarcoplasmic reticulum. So this causes muscle contraction. As we have seen in the previous figure, the calcium ions, they are released by the sarcoplasmic reticulum into the cytoplasm. This is because the action potential travels along the sarcolemma through the T tubules down into the sarcoplasmic reticulum. And now the calcium ions are released. Now these calcium ions, they bind to the troponin component. So this causes conformational changes which moves the tropomyosin away from the myosin binding sites of actin. So here the myosin binding sites of actin filament, they get exposed because of the conformational changes that are taking place here in the actin monomers. So once these are exposed, so the energized myosin is here, okay, that has the ADP plus inorganic phosphate as well as the energy will be there. So this goes and binds to the exposed actin site, causing cross bridging. So you see that cross bridge is formed. Then comes the power stroke. Here the myosin head pulls the actin filament towards the center of the sarcomere, and after this the ADP and the inorganic phosphate, they are released. This generates the force. Next step is detachment and recocking. Now once the ATP, ADP and inorganic phosphate are released, again the myosin is ready for another cycle. New ATP binds to the myosin head and it causes it to detach. Once ATP is bound, it causes it to detach from the actin. Now the ATP is hydrolyzed. It will result in the formation of ADP and inorganic phosphate along with energy. Now this is ready to bind again to the exposed heads of actin, again causing the power stroke. So this is how the actin and the myosin filaments, they'll be sliding one over the other, causing shortening as well as during relaxation, again it comes back to the normal state. So this is how contraction will be taking place, contraction. Let us understand the events of muscle relaxation. This occurs only when the nerve impulse stops. That means there you see that the acetylcholine esterase, it removes the acetylcholine. Okay, and the action potential transfer, it stops. So the first step that has to take place is the calcium reuptake. So this calcium, which is present in the cytoplasm, it has to be pumped back into the sarcoplasmic reticulum. This is through the calcium pumps and it uses ATP. So the calcium ions are pumped back into the sarcoplasmic reticulum while the levels of calcium in the cytoplasm will decrease. Next, the troponin tropomyosin reset will take place. As the calcium ions are gone, the troponin again it changes its shape and tropomyosin it moves back to cover up the myosin binding sites. Previously the myosin binding sites were exposed, so therefore the myosin head could bind here. Now the tropomyosin, as it has moved back, these sites, that is the myosin binding sites, they are covered up, they are blocked. Next, cross bridge cycling stops. So the myosin heads, now they cannot bind as the myosin binding sites are closed here. So cross bridging, this cycle, it stops and the muscle fiber can no longer generate the force. And now the muscle fiber, it relaxes. That is, the filaments, they return back to their resting positions, and the sarcomere, it returns back to its original length, which we call it as the resting length of the sarcomere. Integration of nervous and muscular systems. We see that the nervous system, it is basically controlling the muscle activity by regulating those impulses which are sent from the fibers. The motor unit, it consists of one motor neuron and its muscle fibers. So here, increased impulse frequency, that is known to enhance the strength of the muscle contraction which is taking place. So here, recruitment of more motor units will be taking place, and if that is taking place, it increases even the force that is generated, the force of contraction. So all this is under control, and this ensures that there is smooth, coordinated and purposeful muscle contractions or muscle movements, be it contraction or relaxation. These muscle movements, all of them, they are under control. They are run very smooth and in a very much coordinated way for a purpose. Let us summarize what we have studied. We have seen that there is a coordinated involvement between these two complex systems. That is, we have the electrical events which are taking place as well as chemical events which are taking place. So the nerve impulses which are generated in the axon, they are transferred to the muscle fiber. This is done at the motor end plate, and the muscle excitation is initiated. So excitation, contraction, coupling, that alone converts the signals, that is the chemical signals, into mechanical contraction. So we have already seen sliding filament theory in the previous lectures. So this sliding filament mechanism, that produces the shortening of the muscles and force generation. So all this is integrated process which enables movement, posture, maintenance and even rapid response efficiently. Recommended books for further study include Guyton and Hall Textbook of Medical Physiology, Essentials of Medical Physiology by K. Sembulingam and Prema Sembulingam, or Human Anatomy and Physiology by Elaine N. Mereb et al. Or you can study Review of Medical Physiology by Barrett, Susan, or this is the Indian other one, Textbook of Medical Biochemistry and Physiology by M.N. Chatterjee and Rana Shinde. Thank you.